The name of the experiment is Isolation of Fungus from Spoiled Food. The two types of fungi that are important in food spoilage are yeast and molds. The low pH of fruits is generally unfavorable for the growth of bacteria, but yeast and molds can grow and cause spoilage. Therefore, this experiment is performed to demonstrate the methods that would be implemented for the isolation of fungus from spoiled food so that it can be identified. The requirements for this experiment are potato dextrose agar media, which is also known as PDA, spoiled food sample, petri dishes, weighing machine, autoclave, and laminar airflow. Now, as we start the experiment, the first thing that we would require is distilled water for preparation of the media. So here we have taken 50 ml of distilled water, and potato dextrose agar will be measured now in order to mix with the 50 ml of distilled water. With the help of this digital weighing machine, we are measuring the amount of potato dextrose mixed with the distilled water. As we finish measuring the PDA, the powder of PDA, we mix it with the distilled water that we have already measured. We need to mix it well so that it forms a solution. The media is then sterilized after plugging it with a cotton plug. The sterilization is done in an autoclave. As the media is placed inside the autoclave and the lid properly closed, we shall switch on the power button for the sterilization process to start. After about 15 minutes, we see that the pressure of the autoclave has risen up, and which says that the autoclaving is done. We switch off the power, and at the time we will sterilize or disinfect the area where we will be performing our experiment. The experiment would be performed inside the laminar airflow, so proper disinfection of the laminar airflow is to be done by rubbing the working area with ethanol. The surface is properly clean. We then switch on the blower for a while. The petri dishes which would be used have already been kept inside the hot air oven for sterilization, and now they have been brought out and they also will be placed inside the laminar airflow. Now as we place everything inside the laminar airflow, we will switch on the UV light for some time to allow proper sterilization inside the laminar airflow. By that time, we will also go and check the autoclave. As the pressure lowers down, we will take out the media that has been sterilized within the autoclave. We must be handling it very carefully as the autoclave will be very hot. After taking out the media, we need to take it towards the laminar airflow where we will be performing the experiment. So we will now switch off the UV light and place our media inside it. Before pouring the media into the conical flask properly in order to avoid contamination, after the media is poured into the petri dishes, the petri dishes will be left for a while with the solidifies. Now as the media solidifies, we have taken a spoiled sample of food, that is a spoiled bread. In this spoiled bread sample, we can see some tiny black dots on it, which shows that the boil has been spoiled. The bread has been spoiled, and these tiny black dots are the spores which can grow to produce more mold. So now we will be isolating the mold from the bread and inoculate on the media. The inoculation of the fungi on the media is done by just placing the sample on the PDA. We do not streak it like the way we do in case of a bacterial culture. Fungal specimen from the bread will be inoculated on the media. We will let it incubate for 3 days. The incubation of fungi can be done in normal temperature outside an incubator. So now we will incubate it, and after 3 days we shall observe. On observing the petri dish after 3 days, we see that there are growth of mold on the PDA which we had incubated. Spoilage of bread can be caused by various molds. The molds mostly involved in bread spoilage include Rhizopus, Nuker, Penicillium, Fusarium, or Aspergillus. We will have to perform the lactophenol cotton blue staining in order to confirm the type of mold that has grown on the bread and caused spoilage. So this is how we perform the isolation of fungi from spoiled food.